4 faculty participated in the Cell Collective Working Group. We covered a lot of ground over the course of the semester. I'll give you a quick overview here. In our first meeting, we built a model in Cell Collective using a figure from a journal article found by one of our participants. In our second meeting, we used the model from week 1 to create a lesson that could be used with students. Participants learned how to add pages, panels, content, questions, and more. We also talked about how to design lessons. What are the goals for students, and how does the lesson support those goals? In week 3, participants learned about a brand new feature called course management, which enables faculty to create a course and assign specific lessons to students. This is also where faculty can access student responses to the questions posed in each lesson. Cell Collective has more than 20 pre-built lessons covering a wide range of topics. In week 4, we worked through a lesson titled Exploring Prokaryotic Gene Regulation in the lac operon. Participants learned what it's like for students to build a model and run simulations. We considered what questions are posed to students and why, and we discussed how to modify the lesson to fit the needs of an individual instructor. In week 5, the creator of Cell Collective, Dr. Tom Helikar, joined us. He described his experience using Cell Collective as a platform for authentic student research that ultimately led him to publish with a student. At the end of our conversation, Dr. Helikar provided resources from his course to support participants who want to try something similar. Our last meeting was used to report on progress from the fall semester and plan for spring. Dongfeng didn't have a chance to implement Cell Collective in the fall, but plans to use the lac operon module in her intro bio course in the spring. She also plans to use Cell Collective in an upper-level independent research course where students will model a plant developmental process of their choice. Carlton teaches a 300-level Introduction to Microbiology course where most of his students are pursuing careers in human and animal healthcare. One of his goals is to design and model a simple visual for students to see reduced fitness and realize the cost of maintaining antibiotic resistance phenotypes. Evdokia plans to use the lac and trip operon models in her microbiology course in the spring. She also hopes to use Cell Collective as a platform for students to do research in her Fundamentals of Scientific Research course. Eventually, she'd like to build her own models. Gary introduced modeling to his experimental cell biology class earlier in the semester, using the morphine inhibition of phagocytosis in immune cells that we considered as a group in week 1 and 2. By our last meeting, one student group was working on a final project to examine effects of mutations of the p53 tumor suppressor in Lee-Fermani syndrome. This semester was a lot of fun for me, and I look forward to continuing my partnership with all of these participants.